Okay, good morning, everyone. Welcome to the 2025 Asian Cardiovascular Clinical Research Seminar. This is a session on the cardiovascular protection and regeneration. My name is Hee Wan. I'm honored to moderate this session with my good friend and colleague, Dr. Junyi Yu from Army Medical Center. Today we have four speakers lined up, and our first speaker is also my close colleague at the National Heart Center of Singapore and Duquesne U.S. Medical School. He's the Director of Research and a Senior Consultant, and also the lead director for the Cadence National Platform for Cardiovascular Translation. Today, he's going to talk about cardioprotection. Oops. Cardioprotection in acute myocardial infarction and the challenges and opportunities. Thank you, Derek.
Well, thanks, Yubin, and good morning, everyone. And many thanks to ACCR for giving me the opportunity to present here today. So I will talk on cardioprotection in acute myocardial infarction, challenges and opportunities. So I'll first begin by providing results of a clinical cardioprotection study, which we undertook last year. This is the Petrie study. Then I'll talk about the challenges and opportunities facing translational cardioprotection into the clinic. And then I'll finish off by just sharing on a novel cardioprotective agent which targets mitochondria, MD13 peptide. So we all know that despite current therapy for myocardial infarction, such as revascularization using PCI or surgery, outcomes still can be improved. So these are data from Singapore for heart attack patients or STEMI patients undergoing treatment by primary PCI. There's still significant mortality and morbidity with death rates of 11% and heart failure hospitalization of 7% at 12 months. This is despite therapy given to open up the artery. And therefore, us and others are interested in looking at new treatments to be given at reperfusion to reduce infarct size and prevent heart failure. This is acute cardioprotection. So this is an MRI scan from a patient who's had a heart attack. And you can see on the left, the function of the heart, the left ventricle is impaired here, where there's hypokinesis and thinning of the ventricle. And if we look on the right-hand side, we can see with the gadolinium staining, there is significant infarct and microvascular obstruction. So despite our cardiology colleagues opening up the artery, there's still significant damage to the heart muscle that impairs heart function and can lead to heart failure. So new treatments are needed to reduce the infarct size, prevent MVO, to prevent heart failure. Now, what is the cause of this injury? This is acute myocardial ischemia reperfusion injury. It's multifactorial, which makes it difficult to target and includes inflammation, microvascular obstruction, endothelial dysfunction, calcium overload, reactive oxygen species, and many of these pathways terminate on the mitochondria, which makes mitochondria a nice target for cardioprotection. So in the Cangrelor reperfusion study, in the Petrie study, we wanted to target infarct size as well as microvascular obstruction. So this is the platelet inhibition to target reperfusion injury. So we know that the P2Y12 inhibitors, the antiplatelet drugs that we give our patients who have a heart attack, we give them in the emergency department or in the ambulance on the way to the hospital. Unfortunately, because they're given orally, there's a chance that they would not effectively inhibit platelets at the time of reperfusion because there's a short time from when the patient arrives in the hospital until the artery is opened up. And therefore, in this study, we investigated Cangrelor, which is an intravenous form of the platelet P2Y12 inhibitor given prior to opening up the artery by angioplasty. And we wanted to see or we hypothesized that by offering more maximum platelet inhibition at the time of angioplasty and its potential to decrease cardiomyocyte death, that it may be cardioprotective in this setting. On the bottom right here are some animal studies showing that if you give Cangrelor at reperfusion, you can reduce infarct size significantly. And this Cangrelor seems to mediate its protection through known cytoprotective signaling pathways. And we wanted to use MRI as the endpoint to see if we could reduce infarct size and prevent microvascular obstruction. So this was a multi-center study in Singapore. We recruited nearly 210 heart attack patients undergoing angioplasty. through 6 of the main hospitals, and patients were randomized in the emergency medicine department to receive an IV cangrelor infusion or an IV normal saline infusion. And our primary endpoint was infarct size on an MRI scan performed at day 3, day 4 after hospital admission. And we measured the infarct size, and we assessed microvascular obstructions. Secondary outcomes looked at final infarct size at 6 months, as well as left ventricular remodeling. So this is showing some representative MRI images of a patient, 2 different patients. We did T2 maps to look at myocardial edema, T2 star maps to look at intramyocardial hemorrhage, and we did LGE to look at infarct size. So you can see this patient here has had an anteroseptal infarct, as shown by the late guiding enhancement here, with no microvascular obstruction. Whereas in patient B, you can see a very large infarct in the infralateral region. There's full thickness infarct with this black dense core in the middle of the infarct. This is microvascular obstruction, and this is associated with hemorrhage, as shown on T2 star map here. So this second patient had a much larger infarct with much severe microvascular obstruction and damage. So our aim was to see if cangrelor could reduce infarct size and prevent MVO. So this is the primary data. So acute myocardial infarct size, unfortunately, there was no difference in infarct size between those patients that received placebo or cangrelor. And in addition, at 6 months later, when we looked at the infarct size, there was an overall reduction in infarct size, but in both groups. But again, there was no difference between placebo and cangrelor. And this is quantified here in the table where the infarct size was no difference. Myocardial salvage index, percentage of patients having microvascular obstruction, and percentage of patients having intramyocardial hemorrhage, unfortunately, was not affected or improved by cangrelor. So overall, a neutral study. The studies were presented in the Euro PCR meeting in May last year and were published simultaneously in Circulation. So why did it not work? Our patients are probably too well treated at the moment. In Singapore, they are optimally revascularized as quick as possible. And when we looked in a subset of patients at the level of platelet inhibition in our patients in Singapore, it seems that the oral platelet inhibitors do quite a good job in inhibiting the platelets. And so it may well be in the Singapore setting that cangrelor didn't seem to confer any additional benefits over and above the oral antiplatelet inhibitor. So this sort of leads nicely into the next section of the talk about the challenges and opportunities facing translational cardioprotection into the clinic. For those of you who do research in this area, there's been a huge number of neutral cardioprotection studies looking at many different agents such as nitric oxide, mitochondria-targeted agents, therapeutic hypothermia, beta blockers, ischemic post-conditioning, remote ischemic conditioning, and now cangrelor. And many of these studies show positive results in the preclinical studies, but do not seem to translate into patient benefit in the clinical studies. So why is this? There are a multitude of factors, and these, I just list some of them here. Of course, we all use animal models, mouse, rat, pig. We tend to use juvenile animals with no comorbidities, no complications. Many of us do not use large animal studies such as pig. We always focus on a single target. We usually focus on cardiomyocytes and neglect other cells in the heart, whether it's endothelial cells, fibroblasts, immune cells. And I think many of these studies are single-centered. So one site, maybe 2 sites will do the same drug or the same target. And therefore, maybe we need a more robust approach. So we need more rigorous preclinical assessment using more clinically relevant models, aged models, mouse or rat models with comorbidities. We should consider targeting beyond the cardiomyocyte. That means immune cells and endothelial cells. And then highlighted in yellow here, we should consider multi-center networks. So to make sure that our cardioprotection is more robust. As you know, if you do single center studies, this can be subject to bias. And so if you can then try and do central randomization, central core lab analysis of infarct size, and use a multi-center network, then maybe you can have a much more rigorous preclinical assessment. So that means when you do test your agent in a clinical setting, it's more likely to be effective. So on this topic, we published some guidelines. This was part of our cost cardioprotection network. We published some guidelines to try and improve the chances of translating cardioprotection to the clinic. So of course, you start with your small animal model at a single site. You may consider showing reproducibility at 3 or more sites. Then you may incorporate into your small animal infarct model co-founders, diabetes, age, etc. And then you could move into a large animal model. We would recommend pig. And then you could even consider a large multi-site network randomized control trial using pigs. And then, of course, you go into the clinical evaluation. So in this regard, as part of the cost action, we did set up a network of sites. So these are 8 sites across Europe and the world, including Singapore, where we did a multi-site analysis of 5 sites doing mice and 4 sites doing rats. And we did central randomization from Singapore. We provided this list to the sites. We did centralized core lab analysis of infarct sites in a single lab. This was in Latvia. And so all the samples were blindly sent to them and analyzed. And so it was done as you would do in a clinical randomized multi-center controlled trial. So we wanted to apply this to the basic science setting. So this is the pooled analysis of 5 mice AMI sites. And we looked at the intervention called ischemic preconditioning. For those of you not familiar, this is a rather robust endogenous cardioprotective strategy where you apply brief episodes of ischemia and reperfusion to the heart prior to the infarction. And this has been shown ubiquitously to reduce infarct size. So we use this as our tool intervention to show whether the sites together can show reduction in infarct size. So this shows area at risk across the different sites in sham, control, and IPC. And you can see there's lots of variability across the sites. These infarct sizes were analyzed in a blinded fashion. But here you can see the infarct size in red. This is control compared to preconditioning. There was a significant reduction. So it means that this network of 5 sites, including Singapore and Europe, was able to show ischemic preconditioning, reducing infarct size in a blinded fashion. So this was for mouse. Similarly, in rat, this was 4 rat sites doing infarction, Europe and Singapore again, and we were able to show that ischemic preconditioning was able to reduce infarct size significantly. So this established, at least in principle, that this network of mice AMI and rat AMI sites were able to show ischemic preconditioning, reducing infarct size in a blinded multi-center fashion. And so now this network is available to start testing a novel therapy that someone may in their lab has shown reducing infarct size and now wants to test this in a network, in a blinded fashion. So of course, this would also be very attractive to industry who has a drug that wants to reduce infarct size in their animal studies and now want to test this in a multi-center network fashion. Of course, the next step in cardioprotection is to test this in the pig. So we then formed a network of 5 pig sites. This one didn't include Singapore. This was mainly in Europe. And we caused occlusion to the LAD and reperfusion, and we measured infarct size using 2 methods. One was histology and one was MRI. And then we had a central randomization list. We had central core lab analysis for the histology, central core lab analysis for the MRI. And this shows the overall pool data of the infarct size. We can see that, again, ischemic preconditioning in the pig model was able to reduce infarct size significantly. So again, we seem to establish this pig network that is able to show across 5 sites in a blinded fashion that can be used now for future studies, looking at drugs reducing infarct size in pigs. So on to the final topic of the talk, which is, I guess, opportunities of cardioprotection. Is a novel cardioprotective agent targeting mitochondria. So if I just bring up this schema again, just to remind you, ischemic reperfusion injury is multifactorial and includes factors, inflammation, microvascular obstruction, endothelial dysfunction, calcium overload, oxidative stress. Many of these pathways terminate on mitochondria. So if you can protect the mitochondria during ischemic reperfusion, then you may be able to have a cardioprotective strategy. Now, we know ischemia can induce changes in the mitochondria that include fragmentation or fission, and this can affect its function and predispose it to damage and the lack of ATP production. And this is associated with phosphorylation of the mitochondrial fission protein DRP at serine 616. So given this damaging pathway in ischemic reperfusion injury, we wanted to investigate a peptide called ND13. So what is ND13? ND13 is a DJ1 derived peptide. So DJ1 is a mitochondrial protein that has been shown to be cytoprotective. And our colleagues in Israel and Tel Aviv have designed this ND13 peptide which is a 13 amino acid peptide, which is a mimic of the DJ1, which he had shown, this is Professor Daniel Offen, to be protective in animal models of neurodegeneration as well as stroke. So we wanted to test this ND13 peptide in the heart. We know DJ1 is present in the heart, but its role there is not so clear. So we wanted to see whether administering ND13 in our models of ischemic reperfusion injury could be cardioprotective. So our first step was to administer ND13 to cardiomyocytes isolated from mice, and these mice were Dendra 2 mice. These Dendra 2 mice have fluorescent green mitochondria, so we're able to look at the mitochondria and also look at the fission and fusion in mitochondria. We subjected these cardiomyocytes to simulated ischemia reperfusion, and we treated the cells with ND13. And you can see this dose response with ND13. When we administer ND13, we're able to reduce the number of cardiomyocytes dying during simulated ischemia reperfusion injury. And this scheme on the left just shows when we label the ND13 peptide and we look at uptake of the cardiomyocytes of this peptide, we see that it gets uptake. This peptide is able to be taken up into the cells and confer cytoprotection. So then we wanted to see whether this ND13 can influence mitochondrial morphology. So these Dendra 2 mice have green mitochondria, and these are shows on the left are isolated cardiomyocytes with the green mitochondria. As you can see, they're arranged in a striated fashion according to the myofibrils. And when you subject them to ischemic reperfusion, you can get fragmentation of the mitochondria. As you can see, you get these fragmented round mitochondria. And then if we pre-treat the cells with ND13, we can inhibit this process. So it seems that ND13 treatment reduces cell death, but also can inhibit the mitochondrial fission induced by ischemic reperfusion injury. So what is the mechanism for this? So we hypothesized that it could be through modulating serine 3616 on NDRP. So mitochondrial DRP is a fission protein that's using the cytosol that translocates to the mitochondria in response to stress. And phosphorylation of 616 and 637 can govern its movement from the cytosol to the mitochondria. And in particular, phosphorylation at 616 can induce it to translocate to the mitochondria. Phosphorylation of 637 inhibits its translocation. So we hypothesized that maybe DRP phosphorylation at 616 could be prevented by ND13. So this is showing a phosphorylation of DRP at 616 site, and you can see in those cardiomyocytes given or treated with ND13, you get less phosphorylation at 616. But there was no difference in 637 phosphorylations. So it seemed to be that maybe ND13 is inhibiting siRI-induced fission by inhibiting 616 phosphorylation. So what's the potential mechanism for this? We also looked at ROS. As you know, when mitochondria undergo fission, their respiration is inhibited and they produce ROS. So we wanted to see if we could inhibit the ROS that's produced, and we could see that we were able to inhibit the oxidative stress induced by inomycin by using ND13. So we looked at this Rho-ROC pathway. We know that circulating ROC activity in leukocytes is elevated in heart attack patients. It has been shown that inhibiting this pathway in IRI is cardioprotective. It has also been shown, but not in the heart, that ROC induces IRI-induced cell death by phosphorylating DRP1 at 636. And separately, it's been shown that ND13 can reduce RhoA activity in neurons. So we wanted to see whether ND13, by reducing RhoA activity in the heart, can prevent phosphorylation of DRP at 616. So this is a pathway on the left here that ND13 could inhibit the phosphorylation of serine 616 and inhibit fission. So here we show in our cardiomyocytes that ND13 treatment can reduce RhoA activity. And if we look at the substrate level of RhoA activity, which is MYPT1, we can see that this is reduced also. So this will suggest that ND13 was able to inhibit the RhoA activity, and it may be through this that it inhibits the phosphorylation of 616. When we compared a ROC inhibitor with ND13, we found that both of them could reduce cell death, but there was no synergistic or additive protection. So therefore, if we show less fission with mitochondria with ND13, we wanted to see if we improve respiratory function. So here we show that ND13 can enhance OXPHOS and preserve mitochondrial function in cardiomyocytes following ischemic reperfusion injury. This is using seahorse of isolated cardiomyocytes. And in red here, you can see the effect of IRI, which inhibits or reduces oxidative phosphorylation, as shown by oxygen respiration. And this can be prevented or reduced by attenuated by ND13. And then if we look at the different, this is quantified here, we can see improvements in OCR with ND13 treated cells. And we find that if we look at cytosolic ATP levels, ND13 seems to preserve ATP levels following IRI. So can this be then translated into animal models? So this is the Langendorf model where we take out hearts, we put them on a Langendorf rig, and then we can treat the ND13. We give ND13 at the onset of reperfusion to match the clinical setting where we could give a drug to patients undergoing revascularization by angioplasty. And we circulate ND13 for first 15 minutes of reperfusion to preserve mitochondrial function, and we can reduce infarct size considerably. And then this shows the functional effects. So as expected with ischemia, you get a reduction in function, and then this recovery of function after reperfusion is better in those mice which are given hearts which are given ND13. We then test this in an in vivo setting where we give a bolus of ND13 at the onset of reperfusion, just when we open up the artery, and we find that we can reduce infarct size significantly. And then we use IVIS to measure uptake of the ND13 to make sure it gets into the heart, and we can see that ND13, tagged ND13, goes to the myocardium, the ischemic areas of the heart, showing that it can get to the myocardium and reduce infarct size. So the next step now is to translate this either to large animal models or to consider using our impact network of multicenter sites to see whether this ND13 can really offer robust cardioprotection. So in summary and conclusions, we've shown that rapid platelet inhibition at time of PCI in the Petri study did not reduce infarct size or prevent microvascular obstruction in the STEMI. There are many challenges facing translational cardioprotection, and maybe the use of multi-site AMI networks may improve preclinical assessment and increase the likelihood of translating cardioprotection into the clinic. And then finally, we show the DJ1-derived peptide ND13 inhibits mitochondrial fission and preserves mitochondrial bioenergetics, perhaps via the ROROC DRP616 pathway. Finally, I'd like to thank the fellows in the lab who undertook the work. This includes Sauri Hernandez, as well as Ashwara Prakash, and our collaborator in Israel, Daniel Offen. I'd like to thank the EU Cardioprotection Cost Network for the impact networks, the small and large animal ones. Of course, our funders from Sing Health, Duke NUS, as well as NMRC. And finally, I'd like to thank you for your attention and happy to take questions in the discussion session. Thanks very much.
Thank you, Derek, for the very wonderful overview of your program. And I think we're going to focus our discussion at the end of this session. So let's move on to the next presenter, Jingyi.
Okay, so the next speaker is Professor Zhihua Wang from Fuwai Shenzhen Hospital. His research is mainly focusing on the epigenetics and in the cardiovascular remodeling, especially cardiac aging. So his topic today is MLF1 mediates chromatin remodeling in cardiomyocyte synthesis. So please welcome Professor Zhihua to have your talk. Thanks.
Thank you, Jingyi, for your introduction. Let me share my screen first. It seems I have trouble to share the screen. Okay. Can you see it now?
Yes.
Okay. It's a great honor for me to be here, particularly in this section, hosted by my supervisor, Professor Yibin Wang, when I was a postdoc. Today, I'm going to talk about something about cardiac aging. As we all know, age is the most important risk factors for majority of the cardiovascular diseases, alongside the rapid development of population aging in our society. The treatment of cardiovascular disease will be a big challenge in the future. Cardiac hypertrophy is a common pathological process shared by multiple cardiovascular diseases, and it eventually progresses into heart failure. Of course, the most important pathologies in cardiac hypertrophy is the pathological growth of cardiac muscle itself. And this process is driven by the abnormal gene regulations. Cardiac hypertrophy is also frequently seen in the elderly people. And the aged heart exhibit similar pathologies resembling that pathological cardiac hypertrophy, such as the enlarged cardiac muscle size, inflammation, and fibrosis. However, on the other hand, the pathological cardiac hypertrophy has been considered as an acceleration of the aging process of cardiac muscles. However, it remains unclear whether the mechanism is the same between aging-associated cardiac hypertrophy and pathological cardiac hypertrophy. So in the latest review written by Grumman and colleagues, they proposed the 14 hallmarks of aging, which can be separated into 3 categories, including primary hallmarks, antagonistic hallmarks, and the integrative hallmarks. As you can tell from this figure, 3 out of 5 primary hallmarks belong to the chromatin remodeling, including genomic instability, telomere attrition, and epigenetic alterations. In general, it is well accepted that during aging, the one major feature is the loss of heterochromatin, which means the chromatin structure transforms from a compact structure into looser structures, and this regulation of chromatin structure is tightly related to gene transcription, which largely contributed by the epigenetic modifications, particularly the histone methylations. Until of the histone H3, there are a number of lysine sites that can be methylated, and among these lysines, the K27 is the most special one because it only has one methyltransferase named PRC2 complex, which is a large complex comprised of a number of subunits such as EZ2, ED, and citral. So some previous studies in my lab has established that PRC2 is an important target for non-coding RNAs in cardiac hypertrophy, and the dysregulation of PRC2 function participates in the pathogenesis of cardiac hypertrophy. Beyond the histone methylation, histone acetylation is also an important mediator for the transcriptome reprogramming during cardiac hypertrophy. However, it remains unclear whether there is a mechanism coordinating the histone methylation and acetylation. So at the beginning of my study, we aimed to compare the similarity and the difference between aging-associated cardiac hypertrophy and the pathological hypertrophy. Indeed, the aged heart from 22-month-old mice exhibit hypertrophic phenotypes resembling that observed in the pathological hypertrophy induced by the transaortic constriction, the taxidermy, such as the enlargement in cardiomyocyte cross-sectional area and the fibrosis, but to a much less extent of the in terms of the severity. For example, we do not see significant decrease in the contractile function, and we do not see robust inductions of the heart failure markers. However, at the transcriptome level, we performed an RNA-seq analysis for the aged heart and the pathological hypertrophy heart after TAC, and we see the aged heart exhibit a largely positive correlation with pathological hypertrophy at the transcriptome level. Particularly in terms of the functional clusters, we can see the aged heart share most of the functional terms with TAC-induced hypertrophy. However, there are also a number of unique features for the aged heart. For example, the inhibition of the TCA cycles and the activation of the inflammatory response to type 2 interferon. So we next compared our datasets with public available datasets about heart aging, and we identified 21 genes consistently regulated during cardiac aging. And among these genes, we also narrowed the list and found 4 genes that not only sensitive to aging but can also be reversed by anti-aging administrations. Particularly, we are interested in the factor called MERF1, myeloid leukemia factor 1, whose expression is downregulated in aging and can be reversed by calorie restriction or by the treatment of resveratrol. So suggesting that MERF1, the function of MERF1 may be involved in the mechanistic progress of cardiac aging. So to clarify the role of MERF1 in cardiac aging, we first utilized an in vitro cell model using the immortalized human cardiomyocyte line called AC16. And in this cells, we see that silencing MERF1 can significantly inhibit they enhance the bacterial activity after hydrogen peroxide treatment, and it also blocked the induction of the inflammatory factor, interleukin-1 beta. On the contrary, MERF1 overexpression can significantly enhance, aggravates the H2O2-induced cardiomyocyte senescence in terms of the bacterial activity and the expression levels of the interleukin-1 beta. So suggesting that MERF1 functions as a pro-aging factor. To explore the underlying mechanism, we performed a transcriptome analysis in the AC16 cells with MERF1 knockdown, and we see that more than 800 genes are downregulated after MERF1 knockdown, but only less than 300 are upregulated. So suggesting that MERF1 mainly functions as a transcription activator. Interestingly, the genes sensitive to MERF1 knockdown, they are largely clustered in the chromosomes, indicating there may be some local functions of MERF1 on the chromatin structure. So we performed an ataxic analysis to assess the chromatin accessibility in the MERF1 knockdown AC16 cells, and we see that after MERF1 knockdown, over 10% of the genome are closed, but only 0.5% of the genome are opened. So suggesting that MERF1 may function to facilitate the chromatin openness. And the typical genes subjected to the typical regulation by MERF1 includes a number of inflammatory factors such as interleukin-1 beta, interleukin-4 receptor, and the PDGF receptor. The motif analysis shows that MERF1 mainly regulates genes harboring the purine enriched or the pyrimidine enriched elements. So to further explore the mechanism how MERF1 can regulate the chromatin structure and the gene transcription, we performed an in vitro analysis and say that the genes sensitive to MERF1 knockdown are highly enriched in the histone modifications, particularly the H3K27 trimethylation, as well as its methyltransferase components, including cis12 and the EZH2 here. So we hypothesized that whether MERF1 functions through binding to PRC2 and through the structural docking assay, we see MERF1 has the possibility to bind the PRC2 by accessing cis12 and using co-immunoprecipitation precipitation assays back and forth, and we validated that MERF1 indeed can bind to PRC2 components, including EZH2, cis12, ED, and RBP48, suggesting that MERF1 interact with the whole complex of PRC2. However, no matter we treat the cells with an PRC2 inhibitor, GSK126, or directly silence cis12, we could not see a reverse of the MERF1 hack gene interleukin-1 beta after MERF1 knockdown. So this data suggests that PRC2 may not directly contribute to the regulation by MERF1. So we go back to the histone modifications, and surprisingly, we say that the regulations on K27 acetylation is more special for MERFAN because in the MERFAN knockdown cells, we see a decrease in K27 acetylation, and in the cells with MERFAN overexpression, we see a significant increase of K27 acetylation. The level of histone acetylation is controlled by a balance of the acetylation and the deacetylation processes. So we utilize the two specific inhibitors targeting the acetylation, acetyltransferase EP300 and the inhibitors for the HDAX. And we say that various inhibitors of the HDAX could not reverse the downregulation of interleukin-1 beta by MERFAN knockdown. The inhibitor for EP300 can significantly blunt the enhancement of expression of interleukin-1 beta by MERFAN overexpression. Consistently, we see a similar reverse of the interleukin-1 beta by directly silencing EP300. So suggesting that it is the acetylation process that mediating the function of MERFAN. Next, we ask whether MERFAN directly bind to EP300. So the endogenous immunoprecipitation assay showed that MERFAN can significantly robustly pull down EP300, and by the truncation assays, we verify that MERFAN bind to EP300 through directly interacting with the het domain on EP300. Next, we ask whether EP300 mediating the effects of MERFAN, and whether MERFAN is required for the recruitment of EP300 to certain genomic loci. And we performed the cut and the tag analysis for both EP300 and K27 acetylation. What we see is that after MERFAN knockdown, the chromatin localization of EP300 and the K27 acetylation are both significantly attenuated, and particularly on the genes, directly regulated by the MERFAN, such as interleukin-1 beta, P21, as well as failure marker NPPB. But another marker NPPA is not subjected to the regulation by such kind of regulation by MERFAN. Most importantly, we also performed an RNA-seq analysis for the MERFAN knockdown. We see 16 cells, and we see largely positive correlation between the gene profiles regulated by MERFAN and those regulated by EP300. Basically, the genes upregulated after MERFAN knockdown are also upregulated by EP300 knockdown, and those downregulated are also downregulated here. What's more, the genes sensitive to hydrogen peroxide treatment are significantly reversed by EP300 as well as by the silencing MERFAN. So that's the story of the downregulated genes. Interestingly, we found that in the upregulated genes after MERFAN knockdown, the genes are highly enriched into chromatin loci harboring a number of histone variants. So suggesting that there may be an active compensation process happened in the cells after the disturbance of the chromatin structure by MERFAN knockdown. So taken together, our study revealed factor MERF1 maybe functions as a coordinator for histone methylation and acetylation. Basically, MERF1 may interact with PRC2 to recognize the promoters labeled by K27 methylation, and then recruit an acetyltransferase EP300 to catalyze the K27 acetylation, therefore maintaining the openness of the genes involved in the cardiac aging process. So inhibiting MERFAN function may be a promising strategy to accelerate the aging process of the heart. Just another piece of data, because we all know that the chromatin remodeling during aging is basically an offsite process for that happened in cell differentiation. So we also tested the role of morphine in cardiomyocyte differentiation, and we from stem cells, and we see that during cardiomyocyte differentiation, the expression level of morphine is significantly reduced, and this is accompanied by a significant depletion of the K27 acetylation. And when we knock out morphine in stem cells, we can say it can significantly promote the cardiomyocyte differentiation efficiency. So that's all the story about the morphine in the chromatin remodeling during cardiac aging and cardiomyocyte differentiation. And for this part, we are still working on to finish this project. That's all for my talk. With that, I'd like to thank all the members in my lab contributing to this work. Most of the experiments are done by a postdoc, Lu Jian. And I'd also like to thank my collaborators and grants supporting this work. Thank you for your attention.
Thank you very much, Professor Zhihua Wang. And it's a great talk about the cardiac aging and brought us many new mechanisms. So Professor Yibin, please introduce the next speaker.
Hey, thank you. It is again my great pleasure to introduce the next speaker, Professor Zhang Cai from UCLA. Professor Cai is a professor from biomedical engineering and cardiology at the UCLA, physician scientist, car-carrying cardiologist, really pioneering many of the studies in mechanical sensing and vascular remodeling. Today, he's going to talk to us about the translating biomechanical signals to vascular resilience. John, thank you.
All right, thank you so much, Yibing. And could you hear me?
Yes, yes.
And can you see my slides?
Perfect.
Okay, great. Well, Yibing, thanks so much. And the work we've been doing has been supported by Yibing's legacy to UCLA, the cardiovascular theme. So thanks again, Yibing, for spreading talents across Pacific Oceans. I think being a vascular mechanobiologist, we focus on arteries, but I think the inspiration is to address how the aging process, and during which time this risk factor, hypercholesterolemia, blood pressure, diabetes, and cigarette smoking, can be modified by how could we address chronological agents. And therefore, exercise, we think, provide mechanical signal transduction to mitigate cardiometabolic disorders. I'd like to start with this patient encounter. If you compare two patients, the first patient is 52 years old, presents with exertional chest pain. We heard about the first speaker talking about cardiac protections. In contrast, we have another 94 years old, present with palpitations. Rather than focusing on their metabolic disease, just like to ask you to focus on the blood pressure. And one has a blood pressure of 139/82. The second 145/65. So what's the difference, right? As we age, this compliance curve in terms of aortic volume versus aortic pressure drops, leading to arterial stiffness, and which comes with this progressive pulse rate differences, or the so-called pulse pressure, by subtracting one systolic by diastolic pressure, and that generates the increasing systolic pressure, accompanied by decreasing arterial compliance, and leading to a decrease in diastolic blood pressure. So therefore, I'd like to share with you two potential studies implicating in vascular resilience. So during exercise, and hemodynamic shear stress activates molecular transducers to catalyze vascular protective metabolites. And the second portion will be to share with you our recent finding as to how exercise could modulate neuroimmune interactions to mitigate arterial stiffness. During exercise, I'd like to share with you that hemodynamic shear stress could actually activate a molecular transducer in this region, where you could appreciate atherosclerotic lesions preferentially occur in the curvature as well as branching points. So using a microfluidic model, we can demonstrate this concept and in terms of shear stress acting on the endothelial cells and well known to vascular biologists. And this changes is really controlled by viscosity and a terminology used to address presence of proteinaceous material and cellular components. In terms of clinical translation, blood flow does implicate in vascular homeostasis, as shown in this cartoon, where one could have the so-called pulsatile shear stress in terms of shear forces dyne per centimeter square over time. So PSS abbreviated as pulsatile shear stress. And in vascular biologies, most people use the so-called laminar shear stress, steady flow, to study the activation of cardioprotective oxidants or antioxidants. And so one can appreciate that pulsatile shear stress being cardioprotective in this region, which is the so-called arterioprotective region, and as shown right here. And also you can appreciate the absence of atherosclerotic lesions. And whereas in this region, this is the curvature region where the so-called oscillatory shear stress takes place. So this is the place where, if you look at the blood flow is moving backward and forwards around zero. So the flow is not unidirectional as opposed to pulsatile or laminar shear stress. So it's this region, the so-called disturbed flow, that promotes the activation of inflammatory molecules as well as an NF-kappa B mediated adhesion molecules. And therefore, we hypothesized that during exercise, if you were subject the mice to exercise versus no exercise, one can recondition the atherogenic flow, in this case, oscillatory shear stress, in this curvature and to activate cardioprotective molecular transducer. As shown right here, the activation of flow, this is the endothelial cell in the presence of shear stress we propose that could activate PPAR gamma, which promotes SCD1 expression that converts saturated fatty acid to monounsaturated fatty acids, including oleic acids, which are considered anti-inflammatory for vascular protections. And so using microfluidic model, we can first demonstrate this concept using pulsatile flows versus oscillatory shear stress to isolate potential metabolites using metabolomic approach. And so one can identify, for example, oleic acid, the gray bar indicate the absence of flow, in contrast to presence of flow. So the presence of flow can actually separate the molecular metabolites, including oleic acid and oleic acid, including palmitoleic acids, are being converted by SCD1. And therefore, this inspiration guided us to generate a SCD1 specific knockout model or endothelial specific knockout model and to test the hypothesis. But first of all, we demonstrate the animal model by using visual sonic ultrasound to monitor blood flow in the aortic arch using a mouse model. And so this model allows us to quantify computation of fluid dynamics in the setting of moving boundary of the artery due to stretching and circumferential stretch. But without going through the computational detail, I think I'd like to jump to the next slide just to demonstrate what we discover in that the pulsatile flow in response to exercise could give you these two phenomena. So in the absence of exercise, one can appreciate this disturbed flow region shown by this blue coloration indicating presence of disturbed flow over cardiac cycle during systole at different time components. But in the presence of exercise, this disturbed flow is mitigated, as shown right here. And one can quantify different region of flow component in terms of oscillatory shear index in the greater curvature region versus the lesser curvature. So having this model provide us with quantitative approach to study the mouse model undergoing exercise. And so we generated our EC-specific SCD1 knockout line in the LDL receptor knockout background. And shown right here is the observation and in this region and exposed to disturbed flow versus this region exposed to cardioprotective flow, pulsatile shear stress. And so in the LD region and under no exercise, one can appreciate the endothelial cell and then also the Darby stem for nuclei. But in response to exercise, the activation of SCD1 is stemmed by immunofluorescence versus the absence of ACD1. And likewise, in the descending aorta, one can appreciate the activation of ACD1 during exercise, but then lack of this ACD1 expression in the setting of ACD1 knockout. And this can be further demonstrated by in situ RNA score to demonstrating the presence of ACD1 expressions. Next one, we can show that exercise actually activates ACD1 to mitigate inflammatory marker using VCAN as the example in the lesser curvature. So one can appreciate this diagram. I know it's complicated, but just maybe walk you through quickly in terms of experimental design. So this is the area we're looking at, this area, OSS, exposed to disturbed flow, no exercise versus exercise. And this is the region exposed to cardioprotective flow, no exercise versus exercise. These are the computational fluid dynamic data. But meanwhile, you can appreciate the presence of VCAN1 labeled in red fluorescence and present in the lesser curvature, this region, and then this expression was mitigated during exercise, whereas in the descending aorta, because of the presence of pulsatile flow, cardioprotection flow, VCAN expression is well known to be mitigated by cardioprotective flow in LDL receptor knockout mice in both no exercise and exercise condition. However, the striking part is this area, and you can appreciate that in the absence of ACD1, VCAN expression can be persisted despite exercise. And then in the absence of SDD1 in SDD1 knockout model, you can appreciate SDD1 and VCAN expression even in the area that were protected by pulsatile flow, suggesting that SDD1 expression activated by exercise in this curvature area could be cardioprotective. And next, we perform further study to analyze the metabolites and to illustrate this pathway in terms of oleic acid production and palmitoleic acid production. So LDL receptor knockout mice were undergoing high-fat diet, followed by no exercise versus exercise, and from day 0, day 14 to day 28. So metabolomic study reveal oleic acid as well as palmitoleic acid, as demonstrated from this metabolic pathway that are being upregulated, shown in this bar graph. And during exercise, oleic acid as well as palmitoleic acid, whereas the absence of SCD1 resulted in no activation of this cardioprotective metabolites. And one can also perform SCD1 overexpression to the endothelial cells, indicating the overexpression of SCD1 to the human LD endothelial cell, despite the presence of LPS, lipopolysaccharide induction, this inflammatory marker, CXCL8 or receptor 6A2, all these markers were downregulated in the presence of SCD1 overexpressions. And one can further perform the lysate from the cells that were overexpressed with SCD1, or even the conditioned media to confirm the presence of oleic acid, as well as the downregulation of inflammatory markers such as VCAN1. Transcriptomic data further demonstrate the potential pathway underlying this mitigation of inflammatory marker by way of PPAR gamma, SCD1, and VCAN expression. But without going through the detail, the heat map can demonstrate this association that during exercise, the activation of PPAR gamma expression promotes SCD1, which is associated with a downregulation of VCAN expressions. This model was further supported by using endothelial-specific PPAR gamma dominant negative mice. And you can appreciate that during no exercise versus exercise condition, and this SCD1 expression in the presence of exercise is upregulated, whereas the absence of PPAR gamma resulted in an absence of SCD1 expression. Likewise, the plasma oleic acid that was upregulated in the wild type was no longer upregulated in the setting of PPAR gamma dominant negative mice. Well, so this finding actually has a relationship to what we found in the olive oil and avocado. People ask us, well, what happens when our patients cannot be undergoing exercise? And therefore, you might appreciate that this oleic acid and palmitoleic acid induced by exercise can be found in our diets, suggesting that Mediterranean diets might be the basis to mitigate cardiovascular disease. So this section briefly share with you in terms of exercise mediated mechanical transduction and to reprogram flow recirculation in these regions prone to atherosclerosis. And the changing in this disturbed flow region during exercise may activate molecular transducers such as SCD1 and to catalyze endothelial protective lipid metabolites. All right. Well, this next section is again focusing on exercise, but now I'd like to share with you the inspiration neuroimmune interaction to mitigate arterial stiffness. The initial study was inspired by our colleague Andrea Habernick, as well as Stephanie Dimmler in Germany, demonstrating this neuroimmune modulation might be modulating this region and to mitigate arterial stiffness. Well, this is a complicated slide, but just to share with you this emerging concept, again, recently published by Christian Weber and Hambrecht in Circulation Review, demonstrating this emerging concept of a neuroimmune interaction in the aortic arch and in the abdominal aorta. But without going through the detail, you probably remember Sorsky's seminal work in Science a decade ago, demonstrating that spleen is the source of immune cell to promote atherosclerosis. So that background led to the hypothesis or the goal to demonstrate that exercise modulates sympathetic nerve interaction with immune cell in the aortic adventitia to mitigate arterial stiffness. And so our hypotheses are number one, sympathetic nerves promote aortic adventitia fibrosis by recruiting circulating monocytes to the aortic adventitia, where M1 phenotype induced fibroblast activations. Second, exercise mitigates this sympathetic nerve and macrophage interaction to mitigate arterial stiffness. Well, angiotensin II infusion has been well documented model to promote arterial stiffness, as shown here, with the increase in collagen deposition and also changing aortic stiffness by altering circumferential stretch as well as longitudinal stretch. So with that background, we demonstrate that voluntary wheel exercise running for over 4 weeks, mitigate angiotensin II infusion mediated increase in sympathetic nerve interaction with the adventitia. As shown right here is the experimental design vehicle for 4 weeks, followed in comparison with angiotensin II infusions with exercise. And we focus on abdominal aorta, and the light sheet imaging illustrate the increasing nerve axons in response to angiotensin II, but was attenuated and after exercise, abbreviated as VWR, voluntary will exercise over 4 weeks. And this understanding demonstrate the presence of tyrosine hydroxylase, nerve axons, presence in response to angiotensin II in the aortic adventitia that was mitigated after exercise. And likewise, the extracellular matrix that was accentuated by angiotensin II infusion was attenuated in the presence of exercise model in both thoracic and abdominal aorta. And we further measure pulse rate velocity as a surrogate marker for arterial stiffness and blood pressure measurement using tail vein approach. And to further demonstrate that nerve interaction is implicated in neuroimmune modulation, we can perform celiac ganglionectomy, as shown in this diagram, and to demonstrate that angiotensin II mediated sympathetic nerve interaction can be mitigated after exercise, or can be mitigated after denervations. As shown right here is the Shen group in response to angiotensin II versus ganglionectomy, and one can appreciate the attenuation of nerve axons as labeled by tyrosine hydroxylase. And as a corollary, ganglionectomy also attenuates the aortic thickness and also pulse rate velocity. And next, we found that this interaction was actually being implicated with the beta-2 adrenergic receptor positive macrophages by transcriptomic analysis, and showing that these are all the cells in the aorta, from B cell, dendritic cell, endothelial cell, fibroblast, macrophage, monocyte, neural cell, neutrophil, and T cell. Yet the beta-2 adrenergic receptor expression is prominent in monocytes and macrophages. As shown right here, angiotensin II infusion and promote colocalization of nerve axon with macrophages and CD68s, as shown right here, that was mitigated after exercise. Furthermore, chemical inhibitions, in addition to ganglionectomy, attenuates this nerve monocyte interaction, shown in this diagram. This is the aorta, the adventitia. So the idea is to demonstrate that sympathetic nerve axon interaction with macrophages can be mitigated by ganglionectomy, and also by 6-hydroxydopamine and pharmacological denervations. And you can appreciate the attenuation of synapsins and colocalization with CD68 as quantified by the bar graph. And I think in the interest of time, here just to demonstrate this macrophage nerve interaction to promote fibroblast activation using the cell-cell communication analysis, and also to further demonstrate this experimentally by immunoassays. But in the interest of time, I will probably refer you to our recent publications. Finally, we further demonstrate that pro-inflammatory macrophages are highly expressed in the aortic adventitia, in response to angiotensin II, but that could be mitigated by exercise. In that, macrophage and fibroblasts are the two major drivers to aortic stiffness during angiotensin II infusion, whereas voluntary exercise could reprogram this phenotype to less inflammatory and less fibrotic directions. And so this figure demonstrating that circulating macrophages are recruited to angiotensin II mediated aortic adventitia. And so the bar graph illustrates the present, and if you look at the pink portion, the circulating monocyte in response to angiotensin II, so that portion was reduced in response to voluntary exercise. As a corollary, likewise, monocyte 1 versus monocyte 2. So monocyte 1 phenotype was reduced after exercise. And so this can be further validated by using CCR2 receptor knockout mice to demonstrate that if we were deplete the circulating monocytes, then we could see the reduction of synapsins interaction with monocytes in the adventitias. Finally, I think fibroblasts are implicated in recruiting sympathetic nerve axon in the early adventitia using our neural-related gene ontology study and demonstrating these neurotropic genes and can be activated by the fibroblast. So finally, I like to just to demonstrate that beta-2 arginine receptor is indeed implicated in this neuroimmune interaction. So if you were to give terbutaline beta-2 arginine receptor agonist, and one can actually mitigate the salutary effects of exercise, as demonstrated in this experimental data. So if you look at the bar graph here, the tyrosine hydroxylase staining for nerve axon was present despite exercise due to beta-2 receptor adrenergic agonist. And so the data further demonstrating this in terms of extracellular matrix, ALD thickness, and pulse rate velocity. So finally, I just want to summarize that exercise modulates sympathetic nerve interaction to ameliorate ALD stiffness, and we further use the collagen to this model, and this is the exogen receptor tamoxifen inducible collagen 1A TD tomato lines to demonstrate that angiotensin II mediated sympathetic nerve promotes colocalization of synapsins with macrophages, but not with fibroblasts. So this diagram illustrates the potential mechanism in that exercise could mitigate angiotensin II mediated sympathetic nerve interaction to induce circulating monocytes to express beta-2 adrenergic receptors. And this beta-2 adrenergic receptor activated macrophages promotes fibroblast activations, which further induce axon expression. So sympathetic nerve interactions promotes ALD adventitia fibrosis by recruiting circulating monocytes to the adventitia, where M1 phenotype macrophages induce fibroblast activations, and whereas habitual exercise mitigates the sympathetic nerve and macrophage interactions to attenuate the ALD stiffness. So just to recapitulate, the exercise activates shear stress and to reactivate molecular transducers that might be implicated in cardioprotective metabolites in the endothelial cells. And exercise could also modulate nerve immune interactions and to reduce ALD stiffness. So just to rehash the clinical scenario, remember we talked about the differences between 52 years old versus 94 years old. And I think being a physician, the best medicine is still food and exercise. So I'd like to thank the group for making this possible, and also our cardiovascular team. And I share with Yibing's legacy, and I still think together everyone accomplishes more. And I think that through this teamwork, our training programs have been productive. But of course, it was all Yibing Wen's legacy. So thank you very much. And I know some of you might be attending our Golden Research Conference in Ventura, California. And once again, thank you, Yibing, for creating these amazing opportunities.
Thank you. Thank you. You're being so kind, John. And this is a truly amazing body of work linking the mechanical regulation to both metabolic, but now actually also the sympathetic nerve innervation with the local inflammation by direct connecting with the macrophage. That's truly amazing and make me feel a lot better about running every day. So in the interest of time, we'll move forward to the last speaker of this session, Jing.
So any questions can be raised.
Maybe I'll start. Thank you so much to all the speakers. And I have a first question to Zhipa, and your MEF1 story is really interesting. I wonder whether you actually, in your genetic manipulating mice with the MEF1, what would be your readout for aging, especially cardiac aging in senescence in the cardiomyocytes?
Thank you, Yibin. It's a very important question. We haven't got the in vivo data from the knockout mouse yet, but we just ordered the whole body knockout mice as well as the conditional knockout mice, and the experiments are ongoing now. So hope I can quickly respond to your questions by the updated data. But we do have some evidence of the function of MERF1 in neonatal right ventricular myocytes. And in the cardiomyocyte hypertrophy induced by phenylephrine, we see that knockdown of MERF1 can significantly inhibit the PE-induced cardiomyocyte hypertrophy. So maybe suggesting consistent role of MERF1 in the disease models. Thank you.
But the quick question follow-up is, since the MEF1 involved in most of the factors, either with PRC2 or P300, and they seem to be very generic in every single cell. So do you expect that the same mechanism applies to many other cell type senescence?
Yes, that's another important question. So yes, like PRC2 and EP300, and they are very essential epigenetic factors involved in multiple cell types and disease models. In our observations, we see that in different cell types, the target for MERF1 as well as coordinating PRC2 or EP300 are different. For example, in AC16, we validate that interleukin-1 beta is a direct target for MERF1, but this is not the same case in other human cell lines like HUVEC. But they do have direct targets to regulate through this kind of mechanism. Our hypothesis is that maybe MERF1 is not a typical transcription factors have confirmed elements to bind to regulate in their promoters, but it functions as a scaffold to bridge the epigenetic modifiers like PRC2 or EP300 together with those the real transcription factors. For example, we identified another in another story that MERF1 is a bridge for EP300 and SP1, and care of.
So what you're saying is the MEF1 function in senescence is a cell type specific context.
Yes.
Because of
yes.
Got it. Right.
Thank you.
So maybe we can move on. And I see Derek actually has a raised hand, and yeah, please.
Yeah, hi. A question to Professor Chao Li. I think a very nice talk. I wondered, you know, the Hermes trial by Novo Nordisk is looking at IL-6 antibody in HFpEF patients, and it's a nice link that how the anti-inflammatory effect of the IL-6 antibody could be working through the mechanism of the BCAA in the macrophages. I wonder if you had any comment on this. Thanks.
I think that's possible. Yeah.
Okay, and then one quick question to Professor Sung Sai. So you're saying exercise by causing shear stress can be beneficial, right? But I guess chronic shear stress is bad. So what is it about the exercise-induced shear stress that is good compared to chronic shear stress in the aortic arch? Thanks.
Well, thanks
share
so much for
screen
your
so that-
question.
Okay.
Sorry about the video due to connections. Yeah, but first of all, I already enjoy your talk about ischemic perfusion data. But we thought that there are different types of flow occurring in your circulation system, depending on the branching point versus the descending aorta. So different area of endothelial cells were exposed to different types of flow. But if you were undergoing habitual exercise, we're not talking about competitive exercise that promotes inflammation and oxidation, but rather habitual exercise that recondition the blood flow to mitigate this so-called disturbed flow and inflammatory or atherogenic flow. So if you would mitigate those type of flow during habitual exercise, you might increase the activation of molecular transducer and to catalyze more anti-inflammatory metabolites. Well, obviously that's based on the animal models, and I appreciate I learned from your talk. So maybe the next stage is maybe we should consider clinical trials to elucidate this type of phenomenon. And hope
Thank you very
if
much.
that answered questions.
I also have another follow-up question for Derek. Your ND13 is fascinating, and I wonder if you do clinical before you, I guess, go to clinic, would you be able to demonstrate the cardioprotective effects after the MI is established, let's say a week and 2 weeks later, because that's where you're going to see more patients rather than the emergency room.
Yeah, great question. I think this is a big challenge in the cardioprotection field, especially depends on what you're targeting. If you're targeting this early perfusion injury, then you ideally need to get your therapy on board before you open up the artery. But we are also looking whether there may be additional benefits by giving this chronically. But my feeling, at least for this ND13, it has to be given on board before you reperfuse, which makes it challenging to translate. And I think even before you go to the clinic with this peptide, you have several challenges. First of all, you want to show it can work in a multi-center setting. 2, it can work in a large animal. And then 3, you can deliver it effectively because it's a peptide as well. So you may have to use nanoparticle. But several challenges before you can translate this into the clinic. Thanks.
Sure. I have to ask this question to Tao Li, and our lab has been working on this for many years.
Okay.
And
Yeah.
can you share your insight on exactly what happened to BCAA catabolic defect that triggered all those pathogenic outcome, whether it's in the macrophage or other cells? And let's say in macrophage Y, inflammatory
Yes.
activity actually increased.
Yeah, I actually I have some actual data to show that if the BCA accumulate in the macrophage, it will interact with some proteins that will amplify the inflammatories amplifies the production of some pro-inflammatory cytokines, including IL-6. But I haven't summarized that data yet, so I cannot show that today. So probably I will do that next time.
So you think it's just by the accumulating of the BCA, just the amino acids themselves can
Yes.
cause those effects?
Yeah. Yes. In my opinion, the BCA is very important to the cell. They cannot, if we lack a BCA, the cell will die. But if the BCA the catabolic pathway was damaged, the BCA accumulated in the cell, that will be detrimental to cell. So that I think that's my opinion.
May I ask a question to Professor Hossenlo? Thank you, Professor Hossenlo. I actually read a lot of your pioneering papers, earlier papers about reoxygen species. Regarding the role of ND13 in mitochondrial fission, this is the inhibitor because mitophagy, which is a quality control process for mitochondria, depends on the mitochondrial fission process. Whether inhibition of mitochondrial fission would block the mitophagy and cause the accumulation of damaged mitochondria that cannot be diminished during the disease process. Because in one of my projects, we actually observed a protective role of fission promotion rather than inhibition in in the disease settings of like tach-induced heart failure and ischemia? Thank
A
you.
very, very great question, Professor Wang. I think it all depends on the timing and duration. We would say that you should inhibit efficient acutely at the time of reperfusion, and then it can be cardioprotective. But if you were to inhibit it chronically, then of course you could induce a cardiomyopathy because you inhibit myoefficient chronically, you will inhibit mitophagy, you'll get more damaged mitochondria. So I think it's really the timing. And at least in cardioprotection, I think maybe acute inhibition, transient acute inhibition or mitochondrial fission may be okay, but I think when you're going chronic, then it could be detrimental potentially. Thanks.
Thank you.
Professor Zhuang.
John, please.
Yeah. Okay. Well, again, thanks for all the speakers, but maybe I could just ask a question to Tao Li as well. I was very impressed with your data on CCR2, well, myeloid-specific CCR2 macrophages. You demonstrate that interleukin 6 is implicated. Have you looked at IL-1 beta and whether this increasing IL-1 beta in your myeloid-specific CCR2 may be implicated in activation of cardio and cardiac fibrosis?
Yes, that's a very good question. So actually in our previous study, we found that in the HIF-PEF hearts, mouse hearts and HIF-PEF patients, the IL-1 beta levels was increased in HIF-PEF patients and also in the mouse heart. And in our analysis, we also observed what IL-6, IL-1 beta increased, but that's the extent is not as that high as the IL-6. So we choose IL-6 as a primary target in our project. So, but indeed, the IL-1 beta also increased.
So do you think your CCR2, myeloid-specific CCR2 could be a potential therapeutic target for HAPI?
Yes, I think so. Yeah. I would do more study on that to prove that. Yeah.
Thank you.
Thank you. Yeah.
So Professor Wang, please have a summary of our session.
No. I started, so you should conclude.
Okay. It's my honor to summarize. Thank you very much for all the speakers to support the AHS session. And we have a great talk focusing very related studies in both the heart protection and cardiovascular remodeling. And hopefully our session will be hosted smoothly every year. Maybe we can meet with each other later this year in Beijing. And I think we have a great morning today. Thank you very much for all the speakers and moderators. Thank you. Yeah.
Thank you.
Thanks,
Thank you so much.
everyone.
Thank you, everyone.